Okay, so thinking back to last week, we just got a couple of slides knocked out talking about sensations. Just kind of introduced a little bit about them and the background on them. We talked about the two different types, generals and specials. We're going to start by talking about the first special sense today, smell, and then we'll work through the rest of them. We talked a little bit about some general senses the other day, which included the somatics, but we did not cover the viscerals, and that's okay. I mentioned that that's a little more complicated and stuff I get into a little bit more in like physiology than I do in this class. So like I said, we'll start with the sensation of smell. We're all very aware of our nose as, you know, a young child that is involved with the sense of smell. Fancy term for the sense of smell is olfaction. We've talked about the nose before, thinking back to our lecture on the skull. We talked about the bones and whatnot that form it. We'll talk again about the nose here in a few weeks when we get to the respiratory system, because it's a structure that allows us to breathe, so therefore is a component of our respiratory system. But here we're just going to specifically talk about the structures that allow us to smell. Okay, within the nose are 10 to 100 million of these receptors that allow us to chemically process what we are smelling. The chemicals that we smell are called odorants, and those receptors are found within an area of our nose called the olfactory epithelium. Okay, it is located in the most superior portion of our nasal cavity, covering the inferior surface of the cribriform plate, extending along the superior nasal concha. So thinking back to that lecture on the skull, we have seen these words like the cribriform plate before, the superior nasal conchae. Okay. The cribriform plate is the roof of our nasal cavity. It is the structure that goes horizontally and is formed by way of the ethmoid bone. And we learned that those three little folds on the lateral walls of our nasal cavity are the inferior, the middle, and the superior nasal conchae. Well, this shaded out area here in our picture is where that olfactory epithelium is. And if we see on the right side of the page, we've kind of zoomed in on this box to show the structures that are part of it. Okay, we're going to start kind of down in here talking about the cells that are part of that olfactory epithelium, which include olfactory receptors, supporting cells, and basal cells. Okay, as far as the sense of smell goes, the most important ones are the first ones, the olfactory receptor cells, which also go by the term olfactory sensory neurons. These are the first order neurons that transmit the nerve signal from the peripheral nervous system into the central nervous system. Each receptor cell is a bipolar neuron with an exposed knob-shaped dendrite. So bipolar, remember we learned about those, have the cell body in the center between the two extensions. Okay. If we look at the picture down here at the bottom, our olfactory receptor cells are labeled actually as olfactory sensory neurons, and they are the ones shaded here in purple, kind of this purple bluish color. But you can see the cell body very nicely. There's one extension, there's the dendrite. On the other end is the axon, so dead giveaway that is a bipolar neuron. Okay. On each side of the nose, about 40 bundles of these slender unmyelinated axons of these olfactory receptor cells extend through about 20 foramina or holes in the cribriform plate of the ethmoid bone. And we can see a couple examples of that. Okay, remember the cribriform plate, once again, is the roof of our nasal cavity. It has these very tiny little holes in it, each of which is called an olfactory foramina or foramen. Okay, plural is foramina. But the axons of those various neurons travel through that olfactory, or excuse me, through those olfactory holes, olfactory foramina, and they travel up into the olfactory bulb, which, as we learned in our last lecture, is the very fat end of our first cranial nerve number one. Olfactory cranial nerve is its name. Okay, so those neurons come up and into the olfactory bulb, okay, they then synapse with neurons that you can see here showing like the purple, green, and bluish colors, okay, where they synapse is all once again within the olfactory bulb, okay. So let's read through these 40 or so bundles of axons collectively form the right and left olfactory cranial nerves. Okay, they terminate in the paired massive gray matter called olfactory bulbs, located just below the frontal lobes of our cerebrum. Okay, the part of the olfactory receptors that actually respond to the inhaled chemicals are called olfactory cilium or olfactory hairs. Okay, these hairs are tiny projections on the dendrites of the olfactory receptor cells, and they contain the receptors that are stimulated by odorants, which are the chemicals that have an odor and can stimulate those hairs. So if we look at, and I'll zoom in tremendously here, the very tip of each of these dendrites, you can see the little blue squiggly lines here. Those are the olfactory cilia, the little hair-like structures where the little chemicals that we smell, what we call odorants, combined to trigger them to become excited. Over here on the right is a pretty cool image showing you one of those olfactory receptor cells. Here's the dendrite with the little cilia extending off of it, and that's always going to face down into the nose like so. Okay, over on the right you can kind of see some of those things labeled, but it's a lot harder to show, whereas this one's really cool and you can see things very nicely. Supporting cells also just go by supporting epithelial cells, exactly what their name implies. They're going to support, nourish, and electrically insulate the olfactory receptor cells. They also help detoxify chemicals that may come into contact with the olfactory epithelium. Okay. We can see a couple of them labeled here, and we can see they're the kind of tan shaded color cells over there on the right, or on the bottom, excuse me. And then finally, basal or stem cells, or basal stem cells or basal epithelial cells, which are stem cells located between the bases of the supporting cells. They're the ones kind of shaded here in the light blue. They are the site of cell division, producing the new olfactory receptor cells. So those olfactory receptor cells don't live forever like a lot of other neurons, or I shouldn't say forever, but for our entire lifespan. Those olfactory neurons die off after about a month or so, so they need replaced, which is what those basal epithelials become. They develop and form into those olfactory receptor cells. Okay. We also find within the olfactory epithelium the duct of glands known as olfactory glands or Bowman's glands, which produce mucus, which is going to be produced in this part of the gland and then secreted out at the bottom of the duct where those olfactory cilia are. And the function of that mucus is to help moisten the epithelial surface, and is very important for dissolving odorants so that they can actually make contact with the cilia, and therefore we can actually smell what we're supposed to be smelling. Okay. As far as the images go, don't worry about the two on the right. I do want you to highlight a couple things in these kind of two images here. It's nothing too crazy. Over here on this left image, I want you just to highlight the olfactory bulb and the olfactory epithelium. In this image here, I want you to highlight the epithelium there on the left. And then on the right, highlight the basal epithelial cell, the olfactory sensory neuron, the supporting epithelial cell, the dendrite, and the olfactory cilia. Now, keep in mind where they're like labeled here as basal epithelial cell, or like especially like the olfactory sensory neuron. Make sure you know that's the same thing as olfactory receptor cell. So if you need to write that in parentheses or whatever, might not be a bad idea. Now, very similar to the sense of smell is the sense of taste. Both of them are chemical sensations, and they actually interact with one another. For example, one of the telltale signs of people suffering from COVID is the lack of the ability to smell. That impairs their ability to taste. If you're all clogged up, kind of like I am right now, things don't taste quite as rich. And it's because of that relationship that exists between smell and taste. All right, taste goes by the fancy term of gustation, a chemical sensation, as I've already mentioned, similar to olfaction. There are 5 primary tastes that can be distinguished: sour, sweet, bitter, salty, and then a fun, I believe this is Japanese, umami, which means like meaty or savory. But when you bite into something or you take a drink of something, more than likely what you're gonna experience is not just something that's specifically just sweet or sour or bitter. What you're gonna taste a lot of times is actually a combination of those 5 listed. And now the receptors for taste are located in our taste buds, which I'm sure you've all heard of that term before. Most of our taste buds are found on the tongue, but some are found on the soft palate, which is on the top of our mouth, the pharynx, which is our throat, and even a structure that keeps us from choking called the epiglottis. Structurally, a taste bud is an oval body consisting of 3 kinds of epithelial cells, and it's essentially this kind of same situation that we have inside our nose. You have receptor cells, you've got basal cells, and then you've got supporting cells. The receptor cells, their fancy term is gustatory receptor cells or taste receptor cells, or they can also go by gustatory epithelial cells. We look here at our picture of a taste bud. You can see it's labeled as a gustatory receptor cell. They're the kind of ones that are shown here in the kind of blue shaded color. But they are banana shaped and they have microvilli called gustatory microvilli or taste hairs, okay, which is what they're labeled as down here, microvilli. Little tiny extensions, very similar to those little hairs on our olfactory receptor cells, okay? These are the microvilli that serve as the receptor surfaces for food molecules. So this is where stuff that we taste first binds to those cells to trigger that sensation of taste. Okay. These microvilli project from the gustatory receptor cells to the external surface through what's called a taste pore, which actually you can see is labeled there in the figure. This is an opening in the taste bud. Okay. Gustatory receptor cells are not actually neurons, but they do synapse with the dendrites of first order neurons that are going to make up the first of our sensory pathways. Gustatory receptor cells are a little bit different than the olfactory receptor cells in that they're actually not neurons, but they communicate with the first order neurons. You know, they still essentially do the same thing. They respond to a chemical and ultimately result in action potentials being sent to the central nervous system. Okay. Other cells within our taste buds include these basal cells that you can see here. Also go by the term basal epithelial cells. Those are stem cells that reproduce new supporting cells, which supporting cells also go by supporting epithelial cells are going to surround. You can see they're kind of the clear shaded ones here, the kind of white ones. They surround about 50 of the gustatory receptor cells and actually will develop into them as those receptor cells die off. So essentially the basal cells become the supporting cells and then the supporting cells become the receptor cells. So it's a nice cycle of events that occur there. As we look at this image, where we find our taste buds predominantly is within these projections or kind of elevations on the surface of our tongue called papilla or papilla, however you want to pronounce them. Okay. Fancy actually term for them is lingual. Lingual is a term that always refers to tongue. Okay. And if it's plural, it has the E at the end, so we'd pronounce it papillae. Okay. There's 4 types of them. Filiform, foliate, fungiform, valate. You had to write all those out. Filiform are papillae that have basically kind of a tiny spike appearance, and you can actually see them very nicely over here on the right. But they actually don't even have any taste buds, and instead they contain tactile receptors, which we talked about earlier in the lecture. Tactile receptors we discussed back on this page, which are going to provide us with sensations like touch, pressure, vibration, itch, and tickle. Not that you feel itchy on your tongue, but you can feel like when something's touching the surface, or when you can feel like the texture of food. It's because of those tactile receptors that you're going to find in your filiform papillary or papillae. They increase friction as well, which is going to help the tongue and the food move around to aid with chewing and swallowing. Okay, we find these filiform papillae all over the entire surface of our tongue, and as you can see here, they've just kind of labeled them in the middle of our picture. If you've ever been like licked by a cat or felt the surface of a cat, their tongue is going to feel really sandpapery, and it's because of the amount of filiform papillae that they have. Foliate that you can see here in our image are located in the small trenches on the lateral margins of the tongue. Not really so important because most of them degenerate or kind of die off in early childhood, which means the last two have to have our taste buds because these first two don't. Fungiform papillae are mushroom-shaped elevations scattered over the entire surface of the tongue, contain about five taste buds each, which leaves us with the final type, which it's kind of handy that they kind of have an inverted V shape because the letter V begins the word valate, but they are found at the rear of the tongue, forming that inverted V shape. And up to about half of your taste buds of your entire tongue are located right in this general area. Each of these little bumps is going to contain about 100 to 300 taste buds. So a very significant number. As you can see, we've got a couple of pictures down at the bottom to show some cool stuff. Here's a microscopic view of some taste buds. We've zoomed in on one of them. We can see the different cells. You can see the little hairs. There's the pore that they're sticking out of. Over on the right, we show the surface of the tongue essentially to show the fungiform, which you can see why they get their name. They kind of look like a mushroom, which is where the fungi term comes from. Foliate or the filiform that you can see are also very prominent in there. As we mentioned, those are all over the place on the surface of the tongue. As far as those two bottom images, don't really need to worry about those. This image, I would just say highlight the four papilla that are labeled. And then down here, make sure you know the different cells and stuff that are part of the taste bud. All right, moving on to vision, which is going to be our most complex sensation, both physiologically and anatomically. I would say it is the most complex because there's a lot going on. Obviously, we use our eyes to see, okay, which is what vision is, seeing, okay. But we're going to talk about some other stuff before we talk about the eyeballs, which are the actual structures that allow us to see. We've got some accessory structures that enable our eyes to properly function and do different things for us. And so we'll cover those here for the next couple slides. And then in a couple pages, we'll actually get into identifying the parts of an eyeball. All right, so let's talk about the accessory structures, which include the eyebrows, the eyelids, eyelashes, conjunctiva, the lacrimal apparatus, and then a set of 6 muscles that we'll get into on the next page. Okay, your eyebrows are obviously hairs that are just above our eyes, provide us with the ability to, express facial expression. So like, you raise your eyebrows when you're excited or you're surprised, or you lower them when you're upset. Okay. They also help protect from perspiration. So like sweat's not just going to roll right down into your eyes and other matter as well. Okay. More important for protecting your eyeballs, though, are your eyelids. Now, the first fancy term for eyelids is palpebrae. And if you see that letter E at the end of that word, that means we're talking about both of them. If we're talking about one, it would just be palpebra. But those block foreign objects, protect from excessive light, help with sleep, and blink to moisten our eye, as they act as kind of like a windshield wiper to swipe that water and the fluid across the entire surface. I don't know what the official number is, or the average number of times we blink a day, but it's an incredible number. We can't go very long without blinking our eyes. We all tried as kids to do staring contests, and those don't last very long because we're constantly blinking our eyes. Between the two, your upper eyelid is much more movable than the lower, and it contains in its superior region what's called the levator palpebrae superioris muscle, which is a muscle that helps open that upper eyelid. Its name is very long, levator palpebrae superioris muscle, but its name tells you exactly what it does. Levator, because it elevates or brings upward. Palpebrae, we just learned, is a fancy term for eyelids. And then superioris, so it's going to elevate your superior eyelid. Okay, we can't see it in this picture, but actually here on the next page, we can. Okay, and if we were to go back in time a couple of weeks ago when we talked about the various muscles of the face, it was labeled in, I believe, one of the deep reviews. I kind of mentioned it, but I said we talk more about it later on. Well, here's that point. Okay, The space between the upper and the lower eyelids is known as the palpebral fissure. And you can see that's labeled here with the bracket on the image on the bottom left. The angles, so where the two eyelids essentially meet on the lateral side and the medial side, are called the lateral commissure, just closer to your temporal bones, so on the lateral side. Medial commissures, closer to your nasal bones, so closer to the nose. Okay, on that medial side is where you find a little round bump, and I love this word, lacrimal caruncle. I love that word, caruncle. It's fun to say. But this is a small reddish elevation containing sebaceous and sudoriferous glands. If you don't remember, sebaceous glands produce oil, sudoriferous produce sweat, so it's like an oily, sweaty mixture. Okay? And the whitish material that sometimes collects in your medial commissure comes from these glands. So that's where that kind of crusty stuff that we wake up with is coming from. Okay? Associated with our eyelids and preventing them from sticking together are also tarsal or meibomian glands. They secrete a fluid that helps keep those eyelids from sticking together. They're found in what's called the tarsal plate, which is a thick fold of connective tissue that gives form and support to your eyelids. If you kind of feel for your eyelids, right at the very tip of them is that tarsal plate. It's kind of hard to label, but that's where you find those meibomian or tarsal glands. Also kind of projecting off of that tarsal plate are your eyelashes, which help protect the eyes from foreign objects, perspiration, and the direct rays of sun. Associated with them, and just like any other hair, will be sebaceous glands or oil glands. These are going to be producing that oil and releasing it onto the hair within the hair follicle. We call these sebaceous ciliary glands, and I'm sure if you've ever experienced a sty, you can really appreciate how awful that is. That occurs when one of those glands becomes infected. And it's essentially just like a infected hair or like a zit type situation right on the eyelid. And it's very painful and not super fun to look at either. All right, covering the surface of our eyelids and the surface of our eyeball is the conjunctiva. Conjunctiva is a transparent mucous membrane. It's on the inner surface of the eyelid and the anterior surface of the eyeball, except where the cornea is, which is what we actually look through, and we'll get to that here in a little bit. Okay. Function of the conjunctiva is to prevent your eyeballs from drying out. If you kind of think of like, I always think about like elementary school boys, you know, like some of them like flip their eyelids inside out to scare the girls. Some guys, little boys do at recess. But that layer that you can see is kind of a reddy red shiny material that's on the inside of the eyelid. That is called the palpebral conjunctiva, which makes sense because palpebral refers to eyelid, whereas the bulbar conjunctiva is what covers the surface of your eyeball. Okay, you can see the conjunctiva that's labeled right here. That would be the bulbar conjunctiva. We'll see a picture of the eyeball where that's labeled as well here in a couple minutes. As far as the structures that are labeled here, I do want you to absolutely know those. Some of them include parts of the eyes that we haven't talked about, like the pupil, the cornea, the iris. We're going to get to that here in a second. Same thing with like the sclera, which is like the white part of our eyes. Over here on the right, we show the structures that make up what we call the lacrimal apparatus. Okay, this includes your lacrimal glands or your tear glands, which produce our tears. What we call lacrimal fluid is the fancy term for our tears. Okay, connected to those glands are lacrimal ducts or excretory lacrimal ducts that drain those tears onto the surface of our eyeball. And once again, this is taking place all the time, not just when we're, you know, in a emotional state. We're always producing fluid to help keep our eyes moist. Okay. If we are producing a significant amount of tears, though, we're going to experience, obviously, those tears going down the face, but we'll also have our nose running, and that's because connected to our nasal cavity are a series of ducts that carry those tears from our eyes and into the nasal cavity. That includes a superior and an inferior lacrimal canal. Okay, they are able to pass into those canals through what we call lacrimal puncta, which are two small openings in your eyelids. Okay, those lead to what's called the lacrimal sac, which is found within that lacrimal fossa. If you think back to when we talked about the lacrimal bones, We mentioned the lacrimal fossa. Lacrimal sac then takes those tears and dumps them into the nose through what's called the nasolacrimal duct, or as it's labeled here, nasal lacrimal duct, same thing as nasolacrimal. And those tears are going to then dump into the nasal cavity just below that inferior vena cava, which you can see right there. As far as tears or lacrimal fluid goes, it's predominantly water, but it also contains salt, some mucus, and lysozyme, which actually helps kill bacteria in a really cool way. As far as this image, I do want you to know it as well. And then, make sure you can, keep your head straight on which ducts are which, and, like the order in which the flow of tears occurs. All right, talking now about the movement of our eyes, which is a very controlled and very precise process. It involves the contraction and relaxation of 6 specific, very tiny muscles. These 6 muscles are responsible for moving our eyes in almost any direction, whether that's up, down, side to side, or like rolling your eyes like my wife loves to do with me. But they provide the finest motor control in the body, meaning that they are the most precisely controlled muscles of our body. 4 of them are called rectus muscles. If you think back, we learned that word rectus means straight. So these muscles just basically run straight from the back of the eye and then connect either to the top, to the side, or to the bottom of the eye. Okay, those 4 muscles include the superior rectus muscle, which is what allows you to look up. Inferior rectus allows you to look down. Medial rectus, this is what allows you to adduct your eyes or like look towards your nose. And then the lateral rectus, which allows you to look like to the side, and the abduction motion. And then you've got 2 oblique muscles, which run at an angle to enable you to rotate your eyes. So these are the ones that allow you to roll your eyes. Okay. Let's talk about the connections and how they are associated with our eyeballs. Looking here in our figure A, this is our right eyeball, and we're looking at the lateral side of it. So we've kind of removed part of the face, part of the sphenoid bone, the zygomatic bone, the frontal bone, those kind of things are removed so we can kind of see into that orbital cavity. We get a nice view of the lateral rectus. We get a little bit of a view of the inferior, the superior rectus. We just get a brief little glimpse at the medial rectus. Okay, but those are the 4 recti, and they are all connected to one another in the back of our eye socket and what's called the common tendinous ring. Okay, so that's where they arise, and then they come forward and insert on the actual eyeball, connecting to what's called the sclera, which we'll talk about on the next slide. Okay. As far as the 2 oblique muscles go, the superior oblique also originates back in the deep parts of our eye socket, okay, near that tendinous ring. It then comes forward. We can't really see it here, but it's somewhere back in here, but we do see it right here coming forward. And then there's this little hook-like structure called a trochlea that it bends around and takes kind of like a 90-degree turn. So it enables it to insert kind of on the side of our eyeball. And that way, when it contracts, it pulls the eye like this, which you can actually see really nicely taking place right here. Here's that trochlea. There's the superior oblique, and then you can see it taking its turn going over like so. The other muscle, the inferior oblique, originates on the maxilla, right there on the medial side of our eye socket, and then it comes underneath and just inserts on the lateral side of our eyeball. So the inferior oblique, and you can see it really nicely, just cradles underneath, whereas those other 5 muscles all originate back in the very deep parts of the eye socket and then come forward. Okay. As far as the images go, this is the only one that I want you to know for the exam. And it's just those 6 muscles. So the superior oblique, the superior rectus, the medial rectus, the lateral rectus, the inferior rectus, the inferior oblique. But I also want you to highlight the trochlea because that's associated with that superior oblique. and then the common tendinous ring where the four recti muscles originate. It's really cool to see, you know, this one where you're looking and showing which motions are produced by which muscles. This is a medial or sagittal section showing you the eyeball plus those muscles and the parts of them. Not something I'm gonna ask you to know for the exam, because the one that's here on the next page is the one that I'm going to focus in on the most as far as structures go. All right, despite the fact that your eyeballs are pretty small structures, they're only about 1 inch in diameter. They're very complicated, very, very complicated. A lot goes on with them. I got a lot of pieces to them. And so we'll actually cover the eyeball for 2 slides. So I couldn't fit everything in on this single page. I know probably something, maybe there's some people over there somewhere. All right. Once again, your eyeballs are only about 1 inch or 2.5 centimeters in diameter. Only about 1/6 of your eyeball is exposed. That means the rest of it, the remainder, is recessed, and so it's set back and protected by the orbit, your eye socket. Okay. Each eyeball consists of 3 layers that make up its wall: the outer fibrous tunic, the middle vascular tunic, and then the inner layer, which is known as the retina. Okay, the inner layer is where all the magic happens as far as vision goes, but the other layers are very important as well. Okay, the fibrous tunic also can be described as a superficial avascular layer, meaning that it is on the outside, and then avascular means that it does not have blood flowing through it. Okay, it is divided into 2 kind of regions or 2 different parts: the sclera and the cornea, which were labeled a couple different times. You can see the cornea labeled here on the previous page. I mentioned the cornea on this page along with the sclera. Okay, the cornea is, or excuse me, the sclera, I'll start with that. That's going to form the white of our eye and covers the entire surface except where the cornea is. It is a dense layer of connective tissue made up of mostly collagen fibers and fibroblasts, so there's a lot of strength to it, and therefore it gives the shape to the eyeball, makes it more rigid, protects its inner parts, and this is where those 6 extrinsic eye muscles that we just talked about attach. Okay. Cornea is a transparent coat that covers the iris and helps focus light into the retina. And if you're looking in the figure here, the cornea is labeled where it's kind of shaded blue, but truly you wouldn't be able to really see anything because it's crystal clear. That's what light has to pass through on its way into the eyeball. Okay, the much thicker posterior part of the eye that you can see is the rest of it is that sclera. Okay. If you ever heard of like LASIK surgery, I'm sure you all have. When they perform that procedure, they actually kind of like slice through the cornea and flap it open so that they can do the work on the lens. So that's kind of weird. You can actually watch like videos on that if you're ever interested in that kind of stuff, which some of you might be because you're in this class. But YouTube, you can watch LASIK. It's a pretty cool process how that all goes down. Your cornea actually, I believe, is, if not the most, it's one of the most commonly transplanted structures in the body. Like, when a person dies and they donate their organs, they will snatch that cornea off and donate it to other people, which is really cool. Deep to that fibrous tunic is the vascular tunic, also goes by the term uvea, composed of 3 parts: the choroid, the ciliary body, and the iris. The choroid is labeled down here in our image where it's kind of been peeled away like the sclera was. Okay, just deep to that sclera is where you find the choroid. It's a highly vascular layer, so there's a lot of blood flowing through it, and it's located between the sclera and the retina, so it's behind the retina, and it provides nutrients to that back side of the retina. If you look at this image, okay, it's kind of shown in peach, but you can see these little red and blue dots very frequently in here. Those would be the blood vessels found within that sclera. Okay. The second part of the vascular tunic is the ciliary body. This is the anterior part, and it lies between the iris and the choroid. We'll talk about the iris here in a second. Extends from what's called the oris aorta, which is the jagged anterior part of the margin of the retina, to a point just posterior to the junction of the sclera and the cornea. Okay. Parts of the ciliary body include the ciliary processes, which are protrusions or folds on the internal surface of the ciliary body. Okay, there's also what's called a ciliary muscle that changes the shape of the lens, allowing for near or far vision. So like when you're looking at something small and right in front of you, like when you're reading, or if you're like driving and you're like looking far out in a distance, you're gonna have to change the shape of your lens to focus in on those different things. Okay, pulling on that muscle or relaxing that muscle changes the tightness of what we call the zonular fibers, or as they're also described, suspensory ligaments, which are going to attach the ciliary process to the lens. So if I kind of come down here, here's your ciliary muscle. Okay, coming off of it are the ciliary processes, and then you can see connected to that is the zonular fibers, and those are what connect to the lens. So when the muscle contracts, it pulls on all that stuff, and the lens is going to flatten out. When that muscle relaxes, basically the inverse happens, and it's kind of a cool process. That's how we are able to focus in to, look far away or look really close to one another, to your face. Okay. Your ciliary muscle is a circular band of smooth muscle. And aside from doing that and controlling tightness and shape of the lens, the ciliary body also secretes the fluids that are going to fill the eye that we'll talk about here in just a second. Okay. Let's now talk about the iris. This is the colored portion of your eyeball. This is what can be green or blue or brown or whatever. It is found suspended between the cornea and the lens, and attached at the outer margin to the ciliary process. Okay, the coloring from your eyeball comes from those same types of cells that we learned about earlier in the semester called melanocytes, which we find in our skin. Okay, we learned about melanocytes and that they produce melanin. Well, the same thing is produced here. Melanin, we know, is a protein that gives pigment to our skin and hair. It's also, as we now see, the pigment that gives us our eye color. Okay, if you are brown-eyed, it's because you're producing a large amount of melanin, whereas blue eyes have very little of it, and green eyes have a moderate amount of melanin. Okay. Overall, the key function of your iris is to control how much light is able to travel into your eye through the opening known as the pupil. Okay, the pupil is just the hole at the center of the iris, and associated with the iris are two types of muscles: circular and radial muscles. The circular muscles, if they contract, will cause your pupils to constrict, and that's what happens like when you're exposed to really bright light, so it decreases the amount of light that travels in, which probably would not be the case for you guys right now sitting in this room in Atchison, where I have the kind of front of the room darkened so we can kind of see the screen a little bit better. Your eyes are probably more in a dilated state, where it's because of the radial muscles contracting, allowing more light in. Now, if you flip the lights on or off, those reflexes really are cool to watch. And that's why when you go to the doctor, sometimes they'll shine a light in your face. They're checking to see if those reflexes are working properly. So those are the kind of parts of the middle region of your eye, what we call that vascular tunic. Okay, down at the bottom we can see a really cool picture showing the black pupil there. Surrounding it is the iris, the color part of the eye. There's the ciliary processes, which are associated with that ciliary body, and the muscle that pulls on the lens. And we'll talk about the lens here in a second. But first, we have to talk about the last layer of the eye, the innermost part, which is the retina. And this is where, as I said, most of the kind of crazy processing and functioning takes place within the eye. There are within the retina 120 million rod-shaped cells. We literally call them rods. There's also 6 million cone-shaped cells called cones. These are both photoreceptors. So those are the cells that are going to respond to the light that hits them. Okay. Associated with the retina, which we show here in yellow, are some very important features. The part of the retina right in front of the optic nerve, called the optic disc, is the site where the optic nerve is going to exit the eyeball. This optic disc can be described, and you can see it here in parentheses, as our blind spot, and that's because there's no rods and cones found there. Okay. Probably next time I might do a little test to see if you guys can find that blind spot, but it's really, really cool. Bundled together with the optic nerve are the central retinal artery and the central retinal vein, which feed and drain blood to and from the retina. So this is how blood comes in and out. Okay. The direct center of the back of our eye, where that light is going to most often hit, is what we call the macula lutea, okay, which translates to mean yellow spot. Okay. If I zoom in, you can see is a little bit indented. That indentation is called the fovea centralis. Okay. And this only contains cones and lacks covering, so therefore is a part of our eye that provides us with the clearest vision. This is why when you need to read something, you look directly at it. Okay. And like if you hold a piece of paper in front of your face, you don't hold it to the side. Okay. Look to the side of the room and try to read the board. Can you do it? No. When you want to read something, you have to look directly at it so that all that light is most significantly going to the fovea centralis, to that macula lutea, and which is labeled here in this picture there. And the reason we call them rods and cones, because rods are rod-shaped, cones are cone-shaped. It's as simple as that. Okay, but those are the photoreceptors. Those are the cells that transmit that first original signal towards our central nervous system. Okay, I'm going to tell you without a doubt that this image is going to be on the exam, but I'm not quite ready to highlight it because we aren't done talking about parts of the eye. So we have a little bit left here on the next page to get to. That includes the lens that I've referred to a couple times, along with some spaces where we find important fluids. Your lens is a structure suspended behind the pupil by those zonular fibers of sensory ligaments. It is made up of proteins called crystallins arranged in layers like an onion. I wish we could dissect an eyeball together. We get to do that on campus, where I actually have my students all in one place, and we have a lab where we can do this stuff. It's really cool to get the eyeball into its parts, and you can pull the lens out and you can actually peel it like an onion, which is really cool. Just in front of the lens is a cavity called the anterior cavity, or anterior segment, as it's labeled as here. It is going to contain the type of fluid called aqueous humor, which is more of a watery-like fluid that nourishes the lens and the cornea. Okay. Associated with this anterior cavity is what we call the canal of Schlemm, or scleral venous sinus, which regulates intraocular pressure within your eyes. Okay. Now the anterior cavity is actually divided into two parts: an anterior chamber and a posterior chamber. It's really kind of hard to tell them apart. As far as this image, it's zoomed in quite a bit from like back here. It's kind of harder to tell them apart on that picture we were just referring to. The much larger cavity, which is behind the lens, is the vitreous chamber. Okay, and that's full of a fluid called vitreous body or vitreous humor. And this is probably the coolest thing that you get to play with if you dissect an eyeball. It's a really jelly-like substance, not so much watery, but it's really thick and it likes to stick together. And it's really, really cool. But it's a transparent fluid basically between the lens and then that back of our eye where the retina is. And associated with that big cavity is what we call the hyaloid canal, but it's inconspicuous in adults, runs through the vitreous body from the optic disc to the posterior aspect of the lens. Okay. Here's an image that, like I said, is basically just zooming in at the lens and the structures of the ciliary body that connect to it. Not something I'm going to ask you to know. This is that same image that I showed you on the previous page, but I've zoomed in and made it bigger so you can kind of show those things a little bit more significantly. And this was the one I will tell you to highlight. If you want to highlight them on the previous page, you can, but here it's a little bit bigger, and so I would suggest doing it here. I'll just go through and tell you clockwise what you need to know, and I'll tell you right now, this exam or this picture will absolutely be on the exam. So starting on the bottom, I want you to know these three things: fovea centralis, optic disc, optic nerve. On the left side, the vitreous chamber, hyaloid canal, ciliary process, ciliary muscle, ciliary body, the sclerovenous sinus or canal schlemm. That's what regulates that intraocular pressure. Anterior cavity, you don't need to worry about the two chambers, just the cavity as a whole. And then the cornea, pupil, iris, lens, zonular fibers, ora serrata, retina, choroid, sclera, and macula lutea. Okay, we'll stop here since we're out of time, and we will start with the ear and knock that out where we process what we hear, and we also have receptors there for equilibrium. So we'll get to that on Thursday and wrap this lecture up. Move on from there. All right, guys, have a great rest of your day. I'll see you in a few.